Professor Snap, where we aim to make A-level biology as simple as a cell. So today we're looking at knowing the structure and properties of cell membranes and understanding how models such as the fluid mosaic model of cell membranes or interpretations of data used to develop scientific explanations of the structure of cell membranes. Membranes are vital structures found in all cells. Cell membranes separate the internal cell environment from the external cell environment and holds all the intercellular structures. Membranes also control the exchange of substances from one side of the membrane to the other, as well as acting as an interface for communication. Membranes are partially permeable, and substances can cross through the process of diffusion or active transport. Membranes contain receptor protein which can bind to different hormones and antigens as required. Lipids. So cellular membranes consist of a bilayer of phospholipids. So phospholipids are made of a molecule of glycerol and a phosphate group which forms the phosphate head and two fatty acid tails which makes up the lipid tail. And phospholipids contain two distinct regions, so the polar head and two non-polar tails. The phosphate head of a phospholipid is polar, which means it can therefore interact with polar water molecules. By polar, it means that it carries a charge. The polar head is described to be hydrophilic. The lipid tail is nonpolar. This means it cannot interact with water, which is a polar molecule. So the tail is described as hydrophobic. Phospholipid bilayer forms the basic structure of cell membranes. So the fact that the phospholipid bilayer has parts that are charged, which means they're hydrophilic, and parts that are hydrophobic means that the hydrophilic parts will be facing one way and the hydrophobic parts will be facing the other way. So which means it's going to form a uniform structure around the cell and makes it a good feature that holds the cell together. So the phospholipids bilayers make up cell membranes and they also contain non-lipid components. So proteins are involved with cell transport and communication. And these proteins can either be intrinsic or extrinsic. So intrinsic proteins are in the membrane through the hydrophilic and hydrophobic region. So it's going to pass right through the cell membranes, or just think of it like a tunnel. However, extrinsic proteins are found on the outer or inner surface of the membrane. So they're either on one side of the cell membrane, so maybe the polar side, the hydrophilic side, or it's going to be on the hydrophobic side. So it's like it doesn't go through, it's like on the surface. Okay, but it can be outer or inside. And one way you can remember this is by the fact that intrinsic means right through and extrinsic means you can think of associated with outside, so on the surface. So cholesterol. So cholesterol is also found between phospholipids and its function is very important for cell membranes. And this is a question that is very likely to come up in your exams, as it's been repeated so many times, so keep an eye out for this one. And it has the function of regulating fluidity of membranes. So cholesterol is very useful as it increases the fluidity of the membrane at low temperatures and stops it from becoming too rigid. Cholesterol stops the phospholipid tails from packing too closely together. And in high temperatures, the cholesterol stabilizes the cell membrane at higher temperatures by stopping the membrane from being too fluid. It binds to the hydrophobic tails more and causes the phospholipids to pack more closely. So cholesterol increases the mechanical strength and stability of the membranes as needed, which makes it very crucial. So what are glycolipids and glycoproteins? So glycolipids and glycoproteins are present on the surface of the cells where they aid cell-to-cell communication. Again, this is another important slide that you should really know. And Glycoproteins are proteins attached to a carbohydrate, as you can see in the name. And once again, glycolipids are lipids that are attached to a carbohydrate, which you can also see in the name. And they bind to substances at the cell surface. So some glycolipids and glycoproteins act as cell markers or antigens, which aid cell-to-cell recognition. Fluid mosaic model. So the phospholipid bilayer is often described to follow the fluid mosaic model. And this is because the scattered pattern of the various components makes it look like a mosaic. Explaining the mosaic part of the name, the phospholipids and proteins can move around within the bilayer by diffusion and it is not fixed. Therefore, it is described as fluid, explaining the fluid part of the name. So the membrane is partially permeable, which means not all substances can flow right through the membrane, and small non-polar molecules can pass through the gaps between phospholipids. However, large polar molecules must pass through specialized membrane proteins called channel proteins and carrier proteins. So why is it that polar molecules can't pass through the cell membrane and non-polar molecules can pass through cell membranes? Is the fact that the cell membrane has parts that are hydrophilic and hydrophobic. So the hydrophilic parts, as they are polar, is going to repel the polar molecules, hence they cannot pass right through. However, it doesn't matter if they are non-polar, as they can just pass right through the hydrophilic parts and the hydrophobic parts of the cell membrane. However, we do have an exception with water molecules. Water molecules are polar, which means ideally they should be repelled by the hydrophilic parts of the cell membrane. However, because water molecules are really, really small, they can pass right through osmosis. So it doesn't matter. However, if your molecules are large, they need to pass through specialized membrane proteins like the channel proteins or the carrier proteins. So, models of the cell membrane. So, the fluid mosaic model was first proposed by Singer and Nicholson in 1972. This model evolved over time as we understand more about cell membranes. New features are added to the model with different discoveries. So, then comes the Gotha and Grendel model, which is in the 1920s, and this showed that the phospholipids in the membrane of cells were arranged in a bilayer. So this was the first time that the idea of a bilayer was introduced, and this was proved by the fact that a number of phospholipids extracted from red blood cell membranes were twice the area of the plasma membrane when arranged as a monolayer. However, this model does not explain the location of proteins, how insoluble molecules moved in and out of the cell, which is why this model was disproven. So then comes the Dawson and Danielys model. This model is an improved version which shows the proteins to be arranged above and below the phospholipid bilayer. This model was well supported as the membranes were effective at controlling the movement of substances in and out of the cells. When exploring the electron micrographs, the membranes had two dark lines with a lighter band in between. It was suggested that the darker lines are the proteins and the light band is the phospholipids. However, randomly scattered globular structures were also observed, and further technology explains that proteins were globular, varied in size, and had parts that were hydrophobic. And finally, the model that we follow today by Singer and Nicholson is introduced. So they propose the fluid mosaic model as it is accepted today. According to the model, the membranes were fluid and the globular proteins observed were both peripheral and integral. So freeze-edged electron micrographs showed that proteins extended into the centers of the membrane. So the proteins they observed through the electron micrographs are your intrinsic proteins. So biochemical analysis showed that the membrane proteins are free to move within the bilayer. So that is all from Professor Snap today. Hope you found this video useful. If you have any questions, let me know in the comments below and I will get back to them. And don't forget to like, share and subscribe.